Good morning, everyone. Very excited to be here with you this morning. Thank you to the organizers for bringing us all together and giving me an opportunity to share some of our recent research with the community here. So today I will talk about empowering biomedical discovery through the development of novel multimodal and interactive AI systems that we refer to AI scientists. And I will highlight several projects throughout this presentation that all come with a variety of open source resources, including tools, models, data sets that are linked from our lab website. So with that, let's begin. We have seen over the last several years really increasing integration and exploration of AI models within biomedical discovery processes. And this in part is taking place on two fronts, which the first one is that of AI models changing the way we go about our everyday research activities, and second, increasingly providing insights that might not have been possible with more traditional deep learning models just a few years ago. And I will share some examples of that throughout the seminar. We have seen also early examples of potential opportunities of really more tightly coupling and pairing AI models with drug discovery. So here is an example of that where a phase 2A trial was recently completed showing positive sign of safety and an early sign of efficacy for a compound that was optimized through generative AI models to modulate the target that in part was characterized with the help of AI models as well. So we are hoping that more and more of that will be here in the next several years. So our vision in the group, in the backdrop of these developments over the last several years, is to develop AI scientists. And I use the word AI scientists to refer to an increasing and growing field of collaborative compound AI systems that are able of skeptical learning, reasoning, that can integrate multimodal data with experimental platforms, establish conversational approaches with human scientists, with the goal that eventually these type of models would be able to learn and innovate by themselves. Now, to realize this particular vision, we are operating under this paradigm of AI scientists across multiple scales of biology. And what I want to share with you are several vignettes that will show how we can leverage the AI scientist paradigm and this vision and realizing it by developing foundation models that can help us characterize and better understand the functions and roles of biomolecules, that can then go one scale up and look at the interactions between those molecules and how those molecular interactions can affect the molecular pathways, the ability of patients to respond to a particular type of therapy. All that is taking place in the context of biological environments that can be described and characterized by increasing availability of multi-omics and single-cell data. And fundamentally, eventually, when we look at and characterize the functions of individual molecules, molecular assemblies in the context of cellular environments, all the way gives rise to characterizing the response and the behavior of drugs and therapies in the context of human patients. So let's start with the first part. We'll go from the left towards the right, which is that of molecular modeling and molecular reasoning. So the motivation for this work is observation that as a field at large, we now have AI models that are able to predict the structure of proteins, three-dimensional coordinates of each and every atom in a protein based on primary amino acid sequence of a protein. And for many important classes of protein families, do that at the level of accuracy that matches that of experimental measurements. And so that has been tremendous achievement for the field at large, the ability to have AI models that have this increasing capacity of predicting the structure for every protein. There is, however, another major grand challenge that remains open, which is that of understanding the function or phenotype of those proteins and be able to predict that by considering the structure, the sequence, other type of data we might have around proteins. So can we realize this challenge of being able also to find and characterize the function for every protein in a similar way as we were able to do so field for structures? Why do we want to do that? It turns out that over 20% of human proteins, those directly encoded by genes, still lack functional insights. Practically speaking, that means that when we go to canonical functional databases, curated repositories, and we look at pages for those proteins, that they are relatively empty. We don't really know what they do. 40% of human proteins lack contextual functional insights, meaning that we don't fully understand how the roles and the functions of those molecules vary across biological contexts, such as cell types or disease versus healthy states. And this is a problem that cannot be solved by reasoning or reading literature alone. This is in part because 95% of all publications in life sciences focus on 5,000 most well-studied human proteins, whereas the long tail of the molecules whose behavior we don't understand and how many golden nuggets are buried there. So I want to share with you a model that we've recently developed called Procyon. What Procyon is, it is a foundation-style model, so it is general-purpose AI model that is focused on understanding, predicting, characterizing, and generating protein functions or protein phenotypes, covering broadly 5 knowledge domains, going from molecular processes to disease mechanisms, therapeutic mechanisms of actions, understanding molecular interactions, and substructural units of protein domains, and examples of the kind of tasks that one can ask this model to solve are shown in different rows of this slide. From the user perspective, once Procyon is trained, so human researchers, human scientists can interact with this model through plain natural language promptable interface. Specifically, there are 3 modes that we support in Procyon. So the first mode is that mode of protein retrieval and prioritization. So here, a researcher would describe in plain natural language a particular biological phenotype or protein function they are interested in, and Procyon would read that, and then based on that prompt, it would prioritize and retrieve proteins that would likely satisfy that particular function and play a role in it. The second mode is that of question answering. So here, researchers can engage with Procyon through interactive back and forth discussion, asking questions and the model providing answer about the questions being asked. And what's notable here is that human researchers have the ability to integrate in their input prompt multimodal data, so provide both textual description of the biological function coupled with the amino acid sequence or the structure when available about the molecule they want to learn more about. And the third mode is generative mode, so the model can not only make predictions, meaning predict some form of labels for proteins or generate yes-no answers or retrieve proteins, but it can generate here new textual descriptions. We call this phenotype description generation. Think of it as the model is generating captions, but it's not generating captions for images of cats and dogs. It is generate captions for molecular structures or sequence. And so we call this phenotype generation because it allows us to then go and look at all those stubs of proteins and canonical databases of protein functions that are empty and effectively fill them in by generating these phenotypic descriptions. Okay, so this is how, once the model is trained, you can think about the interface between the AI system and human scientists. So what is within the AI system box? So in the next few slides, I will walk you through the training data set to build Procyon model, the strategy to then train the model using the data set at hand, and also various applications and evaluations of the model. So first, the training data set. So to train Procyon, we created a data set that has the following key components. So first, it had over 33 million protein text pairs. This is called an instruction tuning data set that consists of 33 million tuples, where each tuple is a pair, and it's a pair of protein ID that would be linked then to its sequence or structure, and some textual information in the form of a paragraph or longer that would describe information about that protein as it currently is available across spanning 5 knowledge domains. The second piece of information that is in the protein training data set are 250 million protein sequences that are used to train a component of the model known as protein language model encoder. Then over 800,000 of protein structures that are a combination of experimentally derived high-quality structures from protein data bank with high confidence, with high PLDT values, structures predicted from structure-based models such as AlphaFold 3. And then 50 million full-length papers from the PubMed for the model to be able to understand natural language and converse with human scientists. So this multimodal data set that consists of information about protein and text is then used to train Procyon model. So how do we train Procyon model? We train the model through 3 key training objectives. So the first objective is known as language modeling or next token prediction. The purpose of that objective during model training is to ensure that the model can generate natural language that is coherent and is able to also to understand the language that is encoded in the input prompt of researchers. The second objective is contextual protein retrieval. So this objective technically is achieved through a form of contrastive learning, and the purpose of the objective is to be able to rank and retrieve very highly proteins that have a specific function when it's in the input prompt. So we want to optimize the ability of the model to retrieve in the very top K prediction list those proteins that likely would be positive, most likely be hits for a specific phenotype described in the input prompt. And the third component in the objective function is instruction tuning objective. This is an objective that ensures that the AI model is following human instruction so that it's steerable and controllable. Practically, that means that when a specific input prompt describes the task that the model is asked to solve, the model would actually go and solve these tasks and not approximate version of it or do something completely else. And so training the model through these 3 objectives, we're able then to produce a reasonably large, one of the largest foundation models in biology that has 11 billion model parameters. And now that the question becomes, all this effort that was put into creating the training data set and training these models for several weeks across a large GPU cluster should provide us some type of insights and capabilities that one could not easily achieve or not be able to achieve with more traditional AI models. And so what are those capabilities? There are 4 key capabilities that Procyon has. And then I will also illustrate them with concrete examples. The first one is that of the model being natively multimodal, so it can easily integrate and interleave information encoded in text with information encoded in experimental data in a completely flexible manner. The second capability is that of the model being able to perform zero-shot task transfer. We'll see that in a few slides, but this technical term corresponds to the capability where the model can be used to reason about tasks or execute tasks that it did not explicitly encounter during training time. So it has seen 0 examples of those tasks during training times. That's why it's called zero-shot task transfer. The third capability is that of unbound vocabulary of user input. Practically, that means that we are not limited anymore to reason solely about functions and phenotypes of proteins that are encoded in pre-existing ontological terms, such as gene ontology terms of pre-existing components in standardized terminologies or standardized vocabularies. And this is in part because the model enables free-form conversational input prompt. And the fourth is the model has capability to both make predictions, so retrieve proteins, generate labels and predict labels, and also have generative capabilities of generating textual descriptions of what proteins do. So what we have done first after training the model is what we typically do in machine learning and AI projects, which is to rigorously benchmark the models and specifically perform this type of time machine experiments where we train the model up until on the data generated up until a certain point in time, then ask the model to make predictions, and then use more recently created data to validate the model out predictions. We also have performed a number of few-shot experiments, meaning that the model is asked to predict functions for proteins that were not in the training set, and we can control for also proteins that have either similar structure or similar sequence based on evolutionary constraints. So there are many, many, many plots like this one that we have in the paper. I will not go through all of them. Here's just a single summary slide that shows across different functional categories that corresponds here to different bar plots. We're seeing lift in performance of Procyon over single modality models, such as models that would be trained based on protein sequence alone, protein language models. We're seeing lift in performance over single modality models that are structure-based models alone. Here is an example of that model. It's called GearNet, which is an atom-scale geometric deep learning model that would predict functions based on modeling geometry of structures alone. And we are seeing an improvement over more traditional bioinformatics workflows such as BLAST or related sequence similarity techniques that are often used for functional association and predicting functions based on guilty by association principle. What I'm showing on the right part of the slide here is an interesting analysis that illustrates the benefit of creating these promptable, open-ended conversational interfaces. So here we worked with our collaborators who were interested in FOXC1 and specifically the pleiotropic effects that FOXC1 plays a role in. And so meaning that the functions that they would specifically were interested in would, do not correspond to pre-existing gene ontology terms or reactome pathways or canonical vocabulary items in standardized terminologies, standardized ontologies. And so we were able to show that, well, for FOXC1, which has this pleiotropic functional role, the retrieval of protein, which is on the y-axis percentile, higher value indicates closer to the top of the ranked list of proteins was FOXC1 retrieved when the input prompt contains description of two biological functions or three biological functions or even more complex biological phenotypes. And so that led to benefits that could not be achieved by kind of just simply running the prediction model 2 times or 3 or 4 times if I have 2, 3 functions and then aggregating ranked lists. Okay, let's look at some of the new applications of the model that we have performed with our collaborators after the model was trained. And these are examples of applications that the model did not encounter during training time, was not designed explicitly to be used for, or for its performance to be specifically optimized on these tasks. So there are examples of zero-shot task transfer capability. So here is an illustration of one of those tasks which was using Procyon to characterize the protein structural domains and the binding between drugs against those domains. So the input prompt, which is on the left or part of the slide, shows the input that was provided by researcher, which was the description of the specific protein together with the small molecule compound, miglitol in this case, and some information or an attachment that researcher provide that is information about the structural domains in MGAM. And then Procyon was tasked to prioritize and rank domains in the protein based on how, based on those that were bound by miglitol, and the top one was was the structural domain that indeed is bound by this compound. And we repeated this process for several other proteins and have seen that the rank 1 domain is most often the domain that is indeed bound by drugs. Here's another example, and so here we have been working with collaborators who are studying Parkinson's disease, and they have specific interest in AKNAD1, which was recently implicated in Parkinson's disease. But this is a protein that is empty functional pages. So if we go to SwissProt or UniProt and look at their functional association score, they're low, indicating that we don't know specifically the functional information about those proteins. So we took AKNAD1, and our collaborator described AKNAD1 and what type of function they are interested in knowing whether AKNAD1 plays a role in. And then we used the generative mode of Procyon to generate phenotypic descriptions for AKNAD1 in a conditional manner. And these phenotypic descriptions, we were then able to validate a number of those. Shown here is an example of the predicted involvement of AKNAD1 in the formation of actin cytoskeleton by looking into subcellular maps from the human protein atlas. And so now what we were able to do is to generate this type of protein cards or phenotypic descriptions now for several thousands of human proteins that have empty functional pages in canonical databases. AKNAD1 has relatively low sequence similarity to other proteins that have functional associations. So a more standard approach for predicting functions, which would be to look at other evolutionary proteins with high sequence similarity, look at their functions and transfer that to AKNAD1 would not be successful here. Most recently, we have worked with with 2 labs at Imperial College London and King's College London, and they are studying multiple sclerosis, and specifically we're trying to understand and characterize the disease targets for multiple sclerosis in specific pathologies of MS. And so what they have done is they described pathologies of MS in the form of prompts, which are shown on the left part of the slide. These are the beginning of prompts. Each prompt would be several paragraphs long that describes the observed pathology of MS. And then for each prompt, we use Procyon to rank genes and identify those proteins that would likely play a role in the pathology that is described. And so what we have done then next is that collaborators have looked at 280 postmortem brain tissue samples and stained them and performed RNA-seq analysis for each of those pathologies and use information there on differentially expressed genes in different brains previously associated with pathologies to check to what extent those highly differentially expressed genes in each pathology would correspond to highly retrieved, highly ranked genes by Procyon previously. And so what we can see in the horizontal box plots in the middle of this slide is that across different pathologies, those highly ranked genes with high percentile score, so ranked very close to the top list of the model, were indeed characteristic for the pathology that was later described. So Procyon is a general purpose model. I've showed in the last couple of minutes a few applications. We have 10 more applications where we worked with different experimental groups looking at different molecules, looking at different types of tasks or problems. And so this overall suggests that the model has the capability that make it more generally useful model, but that can be leveraged for a broad number of tasks that can help with characterization and understanding of protein phenotypes. So moving to the next biological scale, which is that of not only understanding the function of individual protein molecules, but look at the interactions that these molecules form and how molecular interactions would affect the behavior of molecules. We'll jump one scale up and look at a model that one of the PhD students, Ada Feng, has recently developed called Atomica. So Atomica is a model that is that is focused on not modeling one molecule at a time, but pairs of molecules or molecular interactions. So Procyon that we just discussed is a model that ultimately what it can do, it will reason about one molecule at a time, like protein, very flexibly about protein, but one protein at a time, right? So, of course, we know that these molecules do not act independently of each other, and so what Atomica can do, it can consider molecular complexes and specifically focus on interaction interfaces between those molecules and model them at the atom scale level. So what does that mean? So that means that what we do is we start going from left to right with an interaction complex, and then we look at the interaction interface of a specific size between two molecules and represent that interaction interface by a graph or a network that is shown in the middle of the slide. So in that graph, we have nodes, which are these circles of different colors representing either molecule 1 or molecule 2. And then edges or links between nodes are edges that carry information about bond lengths, the hydral angle between atoms that are represented by nodes. And then the second level, which is the second row of this graph, contains information about how individual atoms form higher order structures that can correspond to residues or common chemical motifs. So that means that the way the training data set looks like here is that we have over 2 million molecular complexes from Protein Data Bank and Cambridge Chemical Structure Database. And each complex is described by this two-level molecular interaction graph. So the data set is 2 million of this type of graphs that represents atom-scale structure of the interaction interface. So Atomica takes as input this data set of interaction complexes, and it trains a single model through pre-training procedure that can capture the structure and topology of those interaction graphs without explicit human-provided supervised labels. So this is achieved through self-supervised learning, a specific type of objective for self-supervised learning known as one-step discrete diffusion model. But you can practically conceptually think about this as a way to train the model where we hide or block, we mask part of the interaction graph and ask the model to guess what has been what has been masked. So similarly, as text-based large language models are trained for next token prediction, where they mask a specific word and ask the model to predict the word that was masked from the nearby sequences of words. Conceptually, we do we use similar approach here, but this masking is slightly more complex because we don't have a linear sequence of words, but we have this three-dimensional graph structure shape. So we mask an atom, or we mask a chemical motif, or we mask a residue and ask the model to predict it. And that's the process of pre-training the model. An advantage of Atomica is that it is a model that is trained across interactions involving different types of molecules. And that is possible because it's an atom scale model. So the model is trained at the level of atom-atomic interactions. Because of that, we can generalize from modeling protein-protein to modeling protein, small molecule, protein lipid, protein ion, protein nucleic acid interactions because of the underlying shared chemistry. Okay, so after the training of Atomica through the self-supervised objective function, what we get is we get a signature or an embedding for each and every of those 2 million interaction interfaces. And so what you see on the right part of the slide is a visualization of this vast embedding space, or where every point in that embedding latent space represents an interaction interface. And so then we can start exploring and probing that space. So one thing that we have, what we have done by looking at this latent space is we specifically were interested in knowing if by training a single model across different molecular modalities, we can get a better representation of interaction interfaces for those molecules that are currently challenging to model from ML perspective, mostly because the amount of training data are very limited. So what we have done is then explore what is the performance of the model when the model would be trained just on one type of molecular interactions, like protein-protein interactions, versus protein-protein and protein small molecule, or protein-protein and protein small molecule and protein-lipid interactions. And what we can see in this bar plot in the center of the slide is an increasing lift in performance when the model is trained on multiple data modalities. And what is most interesting, and what was most interesting for us, is that the benefit that is the largest for those molecular modalities where the current amount of available data is most limited. So we're seeing the strongest lift in performance in being able to model interactions between the nucleic acids and other molecules when we train a single unified model across these multimodal molecular modalities. And this would broadly be known in the field of AI as scaling laws, which are the increasing capabilities of models as they are trained on larger and more diverse data sets, which is one of key motivations behind training larger models to see this type of emergent or capabilities or the ability to transfer the model across different types of the data. When we have developed the model, we have been recent months using it for a variety of different tasks with the goal of understanding molecular interaction specific disease contexts. But I will show just one example here, which is an example where we applied Atomica to explore the dark proteome. And specifically, we took the Atomica pre-trained model, and then we fine-tuned the model on information about ligand protein interactions with certain properties, and then that gave a version of the model we call Atomica ligand. And that model was used to nominate potential new candidate heme binders in the regions of the proteome that would be known as dark regions, because it consists of many proteins whose roles are completely unknown. And so what we can do with that is that we use Atomica to make predictions, nominate potential heme binders. Our collaborators in the chemistry department over at MIT were then performed experiments to synthesize those proteins and perform functional assay to confirm and test whether those predicted heme binders would actually be heme binders. And so for 5 predictions that the model made, then 5 of those predictions were validated so far, and we are running now multiple second-order experimental campaign of some of these experiments. What is important to note here is that 4 out of 5 predicted heme binders are those that one would not find by looking at common structural motifs of heme binding, which would be a standard or current approach to identify heme binding. And that was important for us to show, because if the model would be nominating only molecules that could be easily found through scanning for the presence of certain structural motifs, there really would not be a big value that the model would provide, given tremendous amount of resources and effort put into developing and training the model. So far we looked at the models that can help us characterize the individual molecules or molecular interactions, and now we will jump one scale up towards understanding how these interactions embed within the broad biological context that can be described by cell types and cell states. So the motivation for this part of the talk first will be outside of biology, just to kind of bring and build the intuition of the need and the importance of considering and modeling biological context through AI models. So if we look at the word apple for a moment, we know that that is a polysemic word. We can grow an apple, we can also buy an apple, and the meaning of this word in specific sequence of words is really determined by sentence context, right? So what we would do, we would look at other words within which this word apple occurs, and that will give us a sense whether the word apple would refer to a fruit, or it would refer to a company or a stock. And so if in the context of molecules and molecular interactions, it's also important to consider this type of biological context, not rather than sentence context. So H2FX is pleiotropic gene, so the meaning, the functions, the roles and biological pathways that it activates or plays a role in can also change and vary across contexts here given by cellular contexts and cellular environments. And so this broad motivation coupled with advances in the last several years, specifically in transformer architectures that can very flexibly model contextual meaning of words in NLP, has motivated really to start asking the questions whether in the context of biology we can develop AI models that would be able to dynamically adjust their context based on which environment, or adjust their outputs based on biological context in which they operate. Think of it as having models that when you ask a model to make a prediction about a molecule, it would not always make the same prediction. But if the question is asked about the same molecule in one context, the output might be different than the same question being asked about the same molecule in a different context. So ideally, the vision here is to really start building these more flexible, adaptive models that would adjust their outputs based on context in which they operate. So we have been developing a suite of models of that type over the last year and a half, starting with a model called Pinnacle. But today I will share with you a more recent model that we have developed called Compass, which has been developed for the purpose of model and help understand and predict immunotherapy and response of patients to immunotherapies by leveraging the idea of contextual learning. Okay, so the motivation for this work is, of course, then observation that immune checkpoint inhibitors, while they have transformed cancer therapy, their response rates to this type of therapeutic modality still remains relatively low. It's somewhat poorly understood and can vary considerably across cancer types. And one of the important questions is, therefore, given the low response rates and considerable heterogeneity of response, is whether one can develop reliable biomarkers that would be able to predict that response from treatment-naive data collected from patients. And so standard biomarkers such as tumor mutational burden or expression levels of key proteins such as PD-L1 have shown poor generalizability from one cohort to the next cohort, as well as from one cancer type to the next cancer type. And so that raises a question whether one can develop more generalizable biomarkers, such biomarkers that would be able to take into consideration the tumor microenvironment of a patient and the context that comes with it in order to improve the prediction of a response for a patient before they are put on the treatment, because that could help with treatment selection and variety of other important decisions that have to be made in clinical trials or treatments. So Compass is a model that addresses this important question. It's a transformer-based model and a specific kind of transformer known as concept bottleneck model. Okay, so what does that mean? So that means that we train the model in two steps, or where the first step is large-scale training of the model through a process we would refer to as pre-training, where we have over 10,000 tumors, and for each tumor, information about the tumor microenvironment from from patients across different cancer types. So it's a pan-cancer data set. And then the model is trained with a transformer Compass model to learn representations of patients' tumor microenvironment. And now the concept bottleneck module allows us to generate these representations or embeddings for patients in a way that they are easily interpretable, meaning that we do not have and obtain some latent signatures of real-valued vectors that are hard to interpret, but we enforce that patient embeddings are expressed through human interpretable immunological signatures and immune concepts, and that is achieved through this concept bottleneck approach. After step one, which is pre-training through concept bottleneck architecture. Step 2 involves taking the model from step 1 that was trained on a large number of patients' data that were not necessarily put on immunotherapies, but on different kinds of cancer therapies. And in step 2, which is fine-tuning step, we take the compass from step 1, and a specific clinical cohort, which can be relatively small, could have only dozens of patients at most in a specific immunotherapy clinical trial. And then the model is asked to leverage part of that information and pan-ups from that cohort to create a classifier. That classifier would then predict treatment response outcome and help with characterizing and explaining, justifying the predicted treatment response at the individual patient level. So how do we train COMPASS model? For pre-training, we use 10,184 tumors across 33 cancer types, where each tumor is represented here by the bulk transcriptional profile of the tumor microenvironment. And we have over 120 curated gene signatures, which are immunological concepts that fit into the concept bottleneck. For fine-tuning and for testing the model, we have collected and be able to work with our collaborators to test the model across 16 clinical cohorts, where each cohort has information about treatment-naive transcriptomes of patients, and then information about longitudinal data, which is the response of patients when they were put on a treatment. And the measure of success here is for a given clinical cohort and given patient in that cohort, given their treatment-naive data, and how well would the model be able to predict a response of that patient to a specific type of immunotherapy. And the better it can predict the response, the higher success of the model, and doing that from treatment-naive information. Okay, so I motivated this part of the talk with the need for generalizable biomarkers that would be able to generalize from one cohort to the next cohort. And so it's a good question, can COMPASS do that? So we've done a number of tests to try to convince ourselves and provide evidence for others as well that the model has indeed better capability in doing so than existing biomarkers. Here's one example of data of such analysis where we have looked at 16 clinical cohorts that we had, and we did an experiment where the fine-tuning process was done on 15 out of 16 cohorts. And then we left out an entire clinical cohort. And so the test is, well, to what extent can a model that has been fine-tuned on 15 clinical cohorts then make predictions for left out cohort number 16, right? That was never encountered, no patient from that clinical cohort, it would be a different clinical study, was ever seen by the model. The goal of that is to be able to see whether there is opportunity and potential for the model to then be used to help with selection of patients for new clinical studies. And so what you can see on the right of this slide, in the rows, we have 22 rows, which are 22 different types of models, including standard biomarkers and different ways of training COMPASS. And we have seen in this cross-cohort transfer, including when we go across small, medium, and large-scale cohorts, much better generalizability of COMPASS than prior models. Here's another example that we have done where we have looked at not only leaving one cohort out, but leaving out all cohorts from a specific indication. So here, for example, then we left out all cohorts with patients in the context of urothelial carcinoma. And so fine-tune the model on cohorts for different indications and then test them on cohorts with urothelial carcinoma. And repeated that process for skin melanoma, for renal cancer, lung adenocarcinoma, and stomach, and to look at the ability of the model to generalize from one indication to the next one. We have done similar tests that I'm not showing here for just a lack of time, where we looked at different therapies and different targets the therapies would have to also test for generalization of models across different specific immunotherapies. Next, we have started applying the model in kind of interesting tasks once we have tested for broad generalizability of the model. And so one example here is that of using the model for patient selection that can help with clinical trial design and clinical trial optimization. So here we work with our collaborators who are designing and running clinical trials for these therapies. And we were able to simulate a trial by seeing, well, what are the patient population that would be selected by the model to be for the trial versus the one that was actually considered, and whether that could help with better patient selection for early phase trials, and we're successful in that. And then the final two results, which I'm very excited about, are results where we looked at COMPASS not solely for predicting the potential response to a treatment, but whether also a prediction of outcomes of those patients. So what you can see here on this slide are 4 Kaplan-Meier curves, which are curves that shows how patients are stratified based on the values of different biomarkers. And so on the far left are COMPASS biomarkers, and then going to the right, we have well-established biomarkers such as tumor mutational burden level or PD-L1 expression levels or immune phenotypes. And what we have seen here, and we have repeated that for several cancer types, is the ability of COMPASS to more accurately stratify patients in a way that would better correlate with overall survival. And then what we can look at next is we can look at what are patients that the model predicts as potential responders to a treatment with longer survival time and goes against the established biomarkers. So what we can see on the left and the right slide are what we call personalized patient maps. So for each and every patient, so on the left here is a patient who was a patient with high tumor mutational burden, PD-L1 levels, and their tumor microenvironment was environment with inflamed immune system. So we would expect, based on established knowledge, that there would be a higher chance of patient responding to this treatment given the activation of immune system in their microenvironment. And indeed, COMPASS predicted with a very high confidence that the patient would respond. And this patient response map that we're seeing on the left allows us to backtrack that prediction to individual immunological concepts, gene sets that underlie those concepts, and all the way back to individual genes that the model relied on when making that prediction. And so this is the benefit of having an interpretable model, because then we can start explaining the prediction made by the model by established immunological signatures, concepts, other gene sets. What was interesting for us is to look at anomalies or unexpected predictions. So on the right is an example of a patient whose tumor microenvironment is really immune desert. So the immune system around tumor is immune desert, so they would likely be non-responders. But for some reason, the COMPASS was very confident that this patient will respond. With 80% probability they would respond. And indeed, the patient did respond to therapy. So why? How can we now explain that? And so we're able to generate this personalized patient map for this patient, and then work with immunologists who looked at the biological pathways concepts that COMPASS relied on when making that prediction that turned out to be correct and would go against expectations. And so we have been able to look at many such examples to then trace predictions back to concepts, back to individual genes, and explore some of the novel mechanisms of resistance to treatments that you were able to confirm in independent orthogonal clinical cohorts. And that is in part possible because of this interpretable capability that the model has. So in the remaining 10 minutes or so that I have, I will switch to another scale, which is that of the scale of modeling not solely tumor microenvironments or tissue-level effects, but really more broadly reason about patients in the context of their entire medical history, in the context of their entire health. So our work in this area in recent years has focused specifically on leveraging the kind of AI models that we refer to in the field as agents. So these agent models, and they're not agents like the agents that we're seeing here in the background, are really agents that are AI systems that can act as agent and be able to perform and execute certain types of analysis autonomously, retrieve data from external databases in autonomous manner, in a self-directed manner. So we have been developing both ecosystems that allow community to create and build AI scientists, as well as creating practical examples of them. So an ecosystem that we have recently built, and it's completely open source ecosystem, is Tool Universe that exists as aiscientist.tools. And the idea here is that Tool Universe allows anyone to build their own AI research assistant from any AI model or AI agent or large language model or large reason model, whether open source or closed source, and do that in a matter of minutes. So how does this work? So if you go to aiscientist.tools, you will see what the process is, and there are many tutorials and examples. Conceptually, there are just two steps, where step one is one selects their favorite large language model, whether that might be Claude, or perhaps you prefer ChatGPT, or perhaps you prefer some open source model. You then connect that model to Tool Universe, and Tool Universe is a universe of scientific tools, which includes software packages, bioinformatics workflows, machine learning models, standard data sets, curated knowledge bases. And then once the large language model is connected to Tool Universe, you are able to interact with those tools in Tool Universe through the interface of the large language model and instruct the model or the AI scientist through that particular conversational interface to perform certain types of analysis. Currently, Tool Universe contains over 600 tools that span 10 categories, which are listed on the right part of the slide here, covering machine learning models, other types of agents, like agents that can do literature research, agents that can create data, scientific packages, workflows, various visualization dashboards, and many other tools. The benefit of AI scientists here and Tool Universe is that it can work with any large language model, reasoning model, including closed form models. That includes Claude, Gemini, Q1 open source model, ChatGPT, GPT Codex. And the way it looks like, if one would connect it with Claude, you get your usual interface in Claude from Anthropic, but there is an option there that allows Claude models to use tools from Tool Universe that we provide. And so immediately you have access, the Claude agent then has access to execute, leverage, and query scientific tools that have been previously validated and established by the community, instead of relying solely on the papers that the model has read itself, which would lead to lots of hallucinations. So how is that possible? How can one build this type of AI scientist, or how can we connect really Tool Universe with these large language models? So the way this is achieved is through new kinds of AI communication protocols. So similar as back in the day, 20 years ago or so, web protocols such as HTTP have transformed internet. Before web protocols, the way internet was just a separate web pages that were not linked to each other. And so it was very hard to perform or do complex searches or explore the complex topics, because it was not easy to navigate from one page to the other. And that was really transformed with the introduction of HTTP protocols and web protocols that have linked pages to each other. And so that led to this ability of not just having a single page, but having these entire web portals or complex websites, and that gave rise to the internet as we know it nowadays. So in AI, what we have seen in the last year or so is also an emergence of AI protocols. And AI protocols specify how AI systems can communicate and talk to each other. So in Tool Universe, we have developed an AI tool interaction protocol, which is a protocol that specifies how a large language model can find, identify a tool to use for specific tasks. So if a task might be perform structural analysis of this protein, a useful tool might be AlphaFold or perhaps Boltz or Chai. And the model has to be aware of the existence of the tool and then know how to call the tool. What are the input parameters, which is specified by the protocol, that would tell the model how to use the tool, how to execute it, and then once it executes it, how to interpret its output and get the response out. So these are the key capabilities that Tool Universe implements and develops so that one can then connect very easily any large language model with these hundreds of tools that exist in Tool Universe. Let me show you an example of an AI scientist that we have recently built that is powered by Tool Universe. So we recently built an AI scientist agent that we named TX agent, which stands for Therapeutics Agent. And it's a broad agent that can help with precision treatments and understanding of precision treatments and optimizing and selecting treatments in the context of individual patient data. So conceptually, you can think of the TX agent as being open-ended conversational approach that would as input take information about the patient, current understanding of the disease to extent it's available, consider different information about the drug, and then it would reason about these inputs by leveraging different types of tools to finally produce an answer, justification for the answer it generated, and its reasoning trace, like a sequence of steps, a sequence of tools it used to arrive at that conclusion. So the tools that TX agent uses are 200 plus tools that cover all drugs developed since 1939 and the clinical insights. So that involves over 150 tools that correspond to API endpoints at FDA, which allows the model to retrieve in real time all the regulatory documents, FDA prescribing card at FDA. The side effect of that is that the model is never obsolete because it's not that we go and download the data and put it on our local data server and we train the model now, so 2 months from now, it's obsolete because it does not know what has happened in the last 2 months. Here, the model in real time, it sends an API request to FDA, get the data from FDA, process that data, and use it to produce an answer and generate response to humans. So it just opens us to completely new ways of how we think about data representations. We have over 50 tools that are tools from Open Targets, a UK-based initiative that has really provides some of the fantastic resources on understanding of ongoing clinical trials, mechanisms of diseases. Those trials are designed for different lines of evidence that support targets for a specific candidate drug. And then we have several dozens of tools from Human Phenotype Ontology and Monarch Initiative that allow the model to connect diseases to broad disease phenotypes. So once we developed TX agent, we have evaluated it in various different ways. And so here's one evaluation strategy that we used, where we looked at FDA drug prescribing cards. And so here are a few pages of screenshots of those cards, which are these 20 to 40 pages PDF documents per drug that they have many sections. And those sections are a review of the drug, information about toxicology, indications, contraindications, the use of drugs in specific patient populations, comorbidities, etc. And so what we tested the TX agent for is to see whether the agent can generate sections of those documents for new approvals. And so what you can see in this plot, we have different groups of bars where each group corresponds to a particular section in this in FDA drug prescribing cards. And then on the y-axis, measure of performance here is accuracy. And we compare TX agent to other large language models that do not use tools and therefore would have to rely on its own internal parametric memory. And that would lead to higher rates of hallucination and poor performance, as well as some of the other models that use tools but very limited number of tools. And TX agent outperform many of those all of those models. The model can be used for personalized treatment understanding. So here on the left, we see a vignette of that where the question that is presented to the model contains more precise information about the patient and their specific situation, and then the model has to reason about that either in a closed-ended multi-choice question answering form or in an open-ended form. And we here again compared the model to TX agent to alternative models and showed substantial lift in model performance. We have then worked with NVIDIA to benchmark TX agent against state-of-the-art reasoning models. So one of those is DeepSeek model that was released a few months ago, and when it was released, it was a big surprise to many of established models before then because it performed so well. And so we were wondering to test whether we can test TX agent to DeepSeek model. And so what the results on the left show is performance of TX agent to 3 variants of DeepSeek models in the DeepSeek family. And TX agent performs much better than DeepSeek models, even though DeepSeek largest model is 2 orders of magnitude larger in its size. So that was very surprising to us. And like, how can we explain that phenomenon? Is it just some kind of an artifact that we are seeing here, or perhaps there's something really wrong with the way we are measuring performance? What we have seen then, analyzing the reasoning traces, is that what happens for those reasoning models that do not use tools or are anchored by entrusted clinically validated insights is that when they are presented with a more complex question, that they would use multiple steps of inference to solve it. They start solving that question, and they might make a mistake in step 3 out of 10 steps. And the problem is, then would just wander off, and step 4 would not be correct anymore, and step 5 is not correct anymore. And so errors accumulate, and the model is unable to backtrack and recover from the mistake made early in the reasoning process. Unlike the DeepSeek, in TX agent, we require that each step is grounded in a validated clinical insight defined as those insights one trusts from and that come from reputable resources. In the context of this project, that would be FDA or open targets. Because of that, the probability of the model to just wander off on a tangent is much lower than in the case of DeepSeek, where each step of reasoning is not grounded in that prior knowledge. And that leads to the better performance of the model, even though in terms of model size, the number of parameters that TX agent has is much less than DeepSeek alone. So key features of these models are 2 things, where A, model being able to reason, perform multi-step inference to solve more complex questions that cannot be solved in a single shot, and be able to use tools to ground its reasoning and retrieve information in real time. Okay, so what are we doing now to test and use TX agent for interesting applications? I increasingly would argue that as we are thinking about training models at scale, like these large-scale general-purpose foundation models, agent systems, we should also think about evaluating models at scale. This is not necessarily happening that much today. We still are seeing prevailing approaches where large models are trained and then we cherry pick 1 or a handful of molecules and then test those. We should test them at scale as well. So we've been thinking about that in the context of TX agent, and have been working with several of our partners to emulate trials for 60 diseases to evaluate therapeutic predictions that the model has generated, where for each of those diseases we're able to identify meaningful endpoint that is a proxy of the disease routinely collected in EHR. Here's just one example of that trial that we emulated for prediction that the model made around protective effect of dapagliflozin against Alzheimer's, which is a drug initially developed for diabetes, and the model predicted and characterized specific patient population that based on the model's reasoning process would get a protective effect from this drug against Alzheimer's. And so to make sense out of that prediction, we look at real-world evidence data and EHR records at Mass General Brigham, where we are able to then create 2 cohorts of patients, control groups, and those that were put on dapagliflozin for the purpose of primary indication for diabetes, and then match those cohorts by sex, by age, by other confounders, and then track the probability of them developing dementia and related phenotypes as a function of time, depending on whether they were put on that predicted drug or not. And the results were very positive and encouraging. We were able then to replicate this result in an EHR system in a different country, which adds more credibility to that potentially being a real effect. But it really shows how we can go from AI models that were not trained on EHR records to seeing potential effects in real-world evidence data and patient populations. We have recently also paired a TX agent with an experimental platform in organoids systems. So here specifically, our collaborators at Harvard Wyss Institute are looking into developing treatments for patients diagnosed with bipolar, specifically those patients who do not respond to standard of care, which is lithium. And so we used the model to generate therapeutic hypotheses, and the testing process involved collecting samples from BD patients and then growing brain organoids from those patients. And then after the organoids were grown, the collaborators administered the predicted drug that the model identified as likely safe and effective for this patient population. So what we can see on the right part of the slide are proteomics readouts of those organoids, and we have different multi-omics readouts. I'm just showing here one type of them, the proteomics readout, where every column in the heat map on the right is one organoid, and then we see in the blue group a control set of organoids from healthy individuals. BD treated group are organoids that were grown from patients diagnosed with bipolar, not responding well to standard of care, and the organoids were treated with the drug predicted by this AI model. And the BD untreated group is the group of our organoids from the same set of individuals that were not treated by using the drug predicted by the model. And we can see the BD treated group, how the proteomic signature has been restored and shifted towards signature that looks more similar to those healthy organoids. And that's corroborated now by different types of omics readouts. So with that, I will conclude my talk. All of the projects across different biological scales that can help us make steps towards the vision of creating increasingly more powerful AI scientist models are made possible by our generous funders and fantastic set of PhD students and postdocs that I'm able to work with. I'm happy to take any questions in the time that's left. Thank you.
Maybe I can go ahead and ask a question. First of all, thank you so much for the presentation. Really, really impressive work. I have one fairly general question. At first, I recently looked into Biomedical, which is another AI scientist model, and I looked into how it was prompted. And there was one section that roughly said, keep in mind you're working with professional scientists, please don't make stuff up. So, and I'm wondering, do you think that a prompt like this is a safeguard? Does an AI scientist know when it's making things up, or what are your strategies to minimize things like hallucinations?
Yeah, that's a great question. Encoding such text in the system prompt of an AI scientist helps, but there are many studies showing that it's not sufficient to get rid of the hallucination. So our approach, unlike Biomedical that you mentioned was not to build a single AI scientist, but instead to build an ecosystem that primarily focuses on tools and protocols that allows tools to be connected with different large language models. And so instead of then, that allows those who are users to, or want to build their AI science models to not be limited to necessarily a single LLM like Claude that the system has been initially built of, but to use any large language model, open source, closed form, or specialized models that might have been trained within your environment that you would trust the most, or perhaps they integrate well with your systems at your institutions. And so just making then these agents, these LLM models effectively use tools without predefining or constraining you to a specific LLM that might have specific limitations in terms of hallucination rates, among others. But there might be other constraints, including constraints that would relate to the ability to potentially send some of the private or confidential information to external systems. That's not a problem for Tool Universe. If somebody would have an internal local model that would allow you to upload because it has been vetted, private or confidential or experimental data, one could still build an AI scientist for that, but it's just connecting Tool Universe to that local model and that would be fine. What we have in Tool Universe, we have a number of guardrails or safety checks that operate at the level of individual tools, not at the level of system prompt. So having a system prompt, putting in the system prompt a single sentence or a handful of sentences, that will have some effect, but it will not really have major effect towards ensuring that the model behaves properly, it uses and interprets the tools the way it should. And so at Tool Universe, we're focusing on a number of different tools, having each tool being vetted by us for correctness. Then we look at questions with respect to how to interpret tools' outputs. Some scientific tools that we use are deterministic. Think of them as calculators. Some scientific tools are non-deterministic. They are stochastic. There are a number of settings where we have multiple tools with overlapping functionalities. And so which tool to select then becomes a non-trivial question and depends on a task. So we might use some tools that are single-cell foundation models. Well, which single-cell foundation model to use? Well, it depends whether, if one would be asking question about immune cells, I would prefer those single-cell model that has been trained or have seen lots of immune cells during training time. So those are all the considerations that promote trustworthiness and reliability of the use of the tools that in no way you can implement and achieve with a system prompt that would apply to all kinds of analysis to the entire institution that uses that AI scientists that are in Tool Universe.
Thank you.
Hi, great talk. A couple of quick questions. One is related to, I saw the resistance models and being able to predict that with some of the inflamed tumor models and things like that. Wondered if you or anybody else is looking at metastases, which is a much more complicated process that happens afterwards. And kind of related to that is things like Parkinson's disease, which basically you have this prodromal time point where you have really not a lot of information about what's really going on at that time, if there's things going on there.
Yeah, that's a great question. In Compass paper, we have some preliminary analysis on data where we separated primary tumor sites from metastatic tumor sites and tested the model in those contexts. It's much more challenging setup. So we are not claiming a tool that Compass solves or addresses that to the extent that would be satisfactory. It's something that we're currently looking into in the follow-up of Compass, specifically focusing on metastatic sites.
For the interest of time, let's continue the session with the next speaker, and then we can continue asking questions and have the Q&A after the next speaker during the coffee break.
Thank you.
Thank you.